Hi, this is Brad Power, and this is the inaugural 2023 meeting of the Prostate Cancer Lab, our weekly meeting. And we're pleased to have with us Mina Nikanjam. I will start off with the disclaimers that we have added to the front end of this so that everyone knows that the information and opinions shared on this website or platform or during discussions and presentations are not intended as medical advice. It is important to consult with a healthcare professional about your specific situation before making any decisions related to your health. And by joining this conversation, you understand and agree that any personal health information you share may be made public. You also understand that HIPAA does not apply. And basically, anything that we say today will be shared in a Zoom recording that will go public, as well as transcript and notes. So if you'd like to not say anything or not share your image, whatever it may be, you have the right to do that now. Otherwise, it will be made public. Okay, with that, I'd like to introduce Mina. Just a bit of a background. Thanks to Allie and her colleagues at CureMatch, we have become very, enamored. We like the idea of drug combinations and the work that Rizal Kurzrock has done that basically says, if you can have multiple drugs that cover the biomarkers that have been identified for you, you're more likely to get a better response. But part of the objection or problem with that drug combination is the issue of there are no randomized clinical trials for some of those combinations, and there's concerns about toxicity. If you have three drugs, particularly with drugs like chemo, they can be additive and they can be harmful to the patient. And so when you add multiple drugs and put them together, the doctors can legitimately be concerned that there may be a toxicity issue and we don't know what's going to happen. And so we were searching around for some strategy for dosing that would complement or control for that issue. And so how can you have a dosing strategy that would be personalized in the same way that drug combinations are personalized and thereby mitigate some of the risks of toxicity? And in a target cancer meeting, I met Mina remotely, virtually. And target cancer is one that Rizal Kurzrock is actively involved in. And Mina was talking about how she had helped with some of the patients that had drug combinations and coming up with a dosing strategy, which is a rare thing. We've been searching and finally found Mina. So that's the backdrop for opening up a conversation today that Mina will lead about dosing strategy. Basically, whenever you're going off-label generally, but specifically around drug combinations.
So I'll share my screen now. Can everyone see this here?
Got it.
Okay. So I'm going to be giving some general discussion about dosing novel drug combinations. So I wanted to start with a bit about my background. It's a little unusual for someone getting into precision medicine. So I started off doing a PhD in bioengineering at UC Berkeley and at UCSF. And I was really interested in coming up with better therapies for cancer. So I was doing a targeted therapeutic for glioblastoma multiforme, one of the worst brain tumors out there. So this is basic science research. I was in a lab, pipetting, doing cell culture. And at the end of my PhD, I really wanted to get a stronger background in clinical aspects and be able to do clinical research and clinical trials. So after my PhD, I went on to do an MD at UC San Diego. And as part of that, I got really involved in clinical pharmacology research. I was doing computer modeling and simulation to assess drug dosing for HIV and oncology therapeutics. So after my MD, did my residency in internal medicine, and then I came to UCLA to do a hematology oncology fellowship. And as part of that, I continued the clinical pharmacology research. For those of you in Southern California, UCLA and UCSD aren't that far apart, but if you hit it at the right time of the day, it's about 2 hours. So I actually did most of my fellowship research at UC San Diego. So in addition to clinical pharmacology, when I was a fellow, I met Dr. Kurzrock and got involved in the precision medicine clinical research. And the task that she gave me was to come up with how do we dose these novel combos. So we did a literature review and looked at data from close to 70,000 patients on clinical trials and looked at how they dose novel combinations. So we looked at small molecules, we looked at antibodies, we looked at chemotherapy, we looked at 2-drug combinations, 3-drug combinations. And from this research, we wrote 4 publications on how we recommended to dose novel drug combinations. Clearly, she liked something I did in my fellowship because she offered me a job at UC San Diego after that. So I was assistant clinical professor for 3 years. I was an investigator on the iPredict and Predict studies and did precision medicine clinical research with Dr. Kurzrock. COVID hit in March 2020, and I was a little bit nervous about seeing patients before we were all vaccinated and wanted to try something new. So I actually went to a pharmaceutical company called Genentech, and there I worked on combination studies. They had a Morpheus clinical trial platform. So I was the breast cancer medical director, but I really missed seeing patients. So I came back to UCSD October 2021, and I currently see medical oncology and hematology patients. I'm part of the Target Cancer Foundation Track Molecular Tumor Board, and then I'm still doing precision medicine clinical research with Dr. Kurzrock. So I've mentioned precision medicine a few times here as part of my background and training. So precision medicine, I'll just put out there, my thought is it's the concept of giving patients individual combination therapies based on the changes or the mutations on the cancer. These are the things that make the cancer grow and make cancer cancer. And that's rather than giving patients treatments just based on the tumor type. In the old days, ovarian cancer got one type of chemotherapy, lung cancer got another type of chemotherapy. But in our precision medicine clinic, everyone gets a unique combination of drugs based on their exact tumor type. I want to start with a case study. This was one of the patients I saw early on when I was an assistant professor at UCSD. So this was a 34-year-old male who was stage 4 melanoma. So this patient with melanoma, if those of you who know melanoma, you know that this used to be one of the worst cancers of them all. People died really quickly. We had no treatment. But in the past 10 to 15 years, there's been a dramatic change in melanoma. These new immunotherapies really have made a big impact in improving outcomes. And in addition to my precision medicine role, I also was expected the melanoma expert too. So I kind of had a split role at UCSD. And many patients with melanoma will do amazingly well with immunotherapy. This patient was not one of them. And there's a minority of patients that still, the immunotherapies just aren't enough. So this poor guy, he had resections, radiation, state-of-the-art immunotherapy, tumor infiltrating lymphocytes. He's been on multiple clinical trials, and nothing was working for this guy. So he came to UCSD to see myself and Dr. Kurzrock for something new. So we put him on the iPredict clinical trial and gave him max therapy. So this was his foundation medicine report. And when we see these reports, we meet in molecular tumor board and come up with a unique combination for the patient's mutation. So based on his profile, he had an NRAS alteration. So based on this, there's a pill called trametinib, which we could give him. CDKN2A, we give palbociclib, another pill. And he had a tumor mutational burden of intermediate. So intermediate or high, they tend to respond better to immunotherapy. So we gave him pembrolizumab. And with these 3 drugs, his first set of scans, he actually had a mixed response. So this heavily pretreated patient who nothing had worked, we actually got some of the tumors to shrink. He ultimately progressed, but if you think about how many therapies this guy had been on, in the prior clinical trial, he progressed immediately. Just having any sort of response was really a success. And you often wonder if you had moved
You
with
guys
match
close
therapy. What was that?
I meant
Was there a question or?
no, my bad. I asked my wife to close the door because they're talking in there,
No,
and
no problem.
I should have been on.
Yeah, so you wonder if this patient had received this match therapy sooner on, first line or second line, maybe we could have had an even better outcome, or he would have had a response that he sustained for some time. So our molecular tumor board, I'll talk about what went on at UC San Diego, but there are many molecular tumor boards out there and they all kind of operate under the same principles. So it's a bunch of physicians. We sit down and discuss what the best combination is for the patients based on the information we have. The tissue testing, liquid biopsies, we put it all together and decide what are the best combinations based on the mutations on the cancer. So the general rules, we look for 2 to 4 drug combinations. If you just use one drug, there's other mutations and pathways for the tumor to grow. More than 4 drugs, it becomes a bit intolerable for the patient. Using 2 antibodies or an antibody and a small molecule to hit a single target tends to improve responses. We've shown this especially to be true for the EGFR pathway and HER2. And after the discussion from Molecular Tumor Board, we come up with a matched therapy option, or we match the patient to a molecularly targeted clinical trial. We keep doing this whenever the patient progresses, and then afterwards, we have phase 1 clinical trials to offer, or even unmatched therapies if we run out of matches to offer. So based on this Molecular Tumor Board, the iPredict study was put together. This is a clinical trial based out of UC San Diego, and it was really aiming to match patients with therapies based on these molecular profiles. And there were several publications and abstracts that came out of the iPredict study, but basically the bottom line is if you had a higher matching score, which is you have more of the mutations with drugs targeted to them, the higher the matching score, you got significantly improved progression-free survival, disease control rate, and overall survival. So patients did better with more matched therapies. So going back to what I did in my fellowship, you have this matched combination. How do you dose them? So this is kind of some general principles that came out of those 4 publications I did with Dr. Kurzrock as a fellow. What we found through our review of all those patients was that the classes of drugs, mTOR, PARP, and HDAC inhibitors tend to be a bit more toxic. Antibodies are less toxic than the small molecule inhibitors and cytotoxic chemotherapy. If you have an overlapping target for a small molecule, this can give you increased toxicity. So if you have 2 drugs both targeting EGFR, that tends to be more toxic than going after separate targets. So for small molecules, we start most of them at 50% dose. Many of the antibodies, because they're better tolerated, can be started at full dose. mTOR, PARP, and HDAC inhibitors are very cautious. We'll often start them at 25% dose or even lower. So we make a dosing recommendation. We see the patients every week in clinic. We get labs. We see how they're doing. And then we slowly titrate the drug up, each of the drugs, so we get to a level where we're treating the cancer well, and then the patient's not getting sick. If you start too low, it doesn't work. If you start too high, the patient gets sick and you have to stop therapy. So it's a fine line in starting at the right level and going up slowly so the patient's not getting sick and you can continue therapy and you're also treating the cancer well. So the last slide I had, I think Brad had asked me to talk about how do you actually get these drugs to the patients. This was a challenge sometimes. So we're giving a lot of these drugs off label. So if you put this through insurance, they tend to decline it. No matter how many people you talk to on the phone, of peer-to-peers, the insurance company just says no way and keeps issuing denial letters. We had a great team at UCSD, which worked on medication acquisition and was able to get the medication for most of the patients free of charge using patient assistance. Most of the patients in California, particularly Southern California, we saw them and treated them at UCSD. For patients who are on all pills, UCSD, we monitored the therapy and prescribed the pills. For patients requiring IV therapy, especially those outside of California who were getting them once a week or every other week, it was hard for them to come to UCSD for every single treatment. So we tried to partner with outside oncologists. And I got a wide variety of responses. So for patients who are on later line therapies with no good options, many physicians said, okay, we'll give this a try, even though it's something new, and we'll set up the infusion. But a lot of the physicians actually refused, and no matter how much you talk to them, they just wouldn't give you the treatment. So the patients were very proactive in finding different oncologists to try to make it work for themselves, and we were able to get pretty much all the patients the match therapy that wanted to. And the final thing I wanted to say for this initial part is that Kaiser Permanente patients offered a very unique challenge because I can't actually prescribe meds at Kaiser. And if the Kaiser physician orders it, the way their system works, it's approved. So I couldn't actually get denial letters to get the patient's patient assistance for the Kaiser patients. And when they came to see me, they had to pay out of pocket. So it really depended on the doctor. If the Kaiser doctor was on board, things went really smoothly. We got all the drugs, the patients were treated, everything was managed well. If the Kaiser physician said no, the patient either had to find another doctor within Kaiser, or many of them actually switched insurance so that they could come and see me. So if this was something that was being done in November, it was kind of easy to switch. But if it was a February, they were seeing as a consult. That's a long time to wait to come and see me for match therapy. So that was a unique challenge with getting meds. So that was kind of an overview of what we do in molecular tumor board, how do we dose these drugs, and how do we get the drugs to the patient. So I'll pause there and open up for questions and discussion.
Thanks, Mina. I will moderate, and if we could use the hand raising feature. And there have been a number, Mina, you might want to look in the chat if you can see them. There have been a number of questions there, but I'll just call on everyone in sequence. Rick had a few questions. Why don't we start with Rick?
Hi. You mentioned TILs in a previous slide, but I don't know what that means.
Tumor infiltrating lymphocytes.
Right.
The concept is you harvest these lymphocytes and then expand them and then inject them, give them back to the patient. They actually do it in kind of a stem cell transplant where they suppress the immune system and then infuse the TILs. It's still, I don't think it's FDA approved yet. It's still in clinical trials, but for melanoma, at least, there's been a lot of promise with the TILs.
Okay. So that
it's a fancier, newer kind of immunotherapy.
Well, that's kind of interesting. So what is, a lot of prostate cancer patients, we don't have TILs. So what do we do in that case? Right.
Yeah, I think that kind of speaks to the whole question of immunotherapy. So the immunotherapies in general kind of work well for certain cancer types, and for other cancer types, we're still kind of looking for more things that we could do. One of the things I mentioned on the slide, the tumor mutational burden of intermediate or high, the patients tend to benefit from immunotherapy. Also PD-L1 expression, they benefit, MSI high, but I have some patients that come to me and really want immunotherapy and they don't have any of the markers. And if you look at the studies, you don't have any of those markers, these immunotherapies just don't work well. So there's some people that are doing research to see, is there a way we can stimulate the immune system to get people to generate these tumor infiltrating lymphocytes and get them to put up a better immune response, but it's still under investigation.
Yeah, that I was going to focus on that as well, is that it seems like immunotherapies can be a great source for durable responses. And it's since we're very biased towards diagnostics, what are the tests that would indicate whether you're going to be a likely responder to an immunotherapy? And you just sort of ticked off the ones. I guess it's MSI, tumor mutational burden, and then PD-L1 likely. The other one that we've identified, and again, Rick's the expert in this, but it's if you can do spatial analysis and see whether the T cells are mixed in with the cancer cells or whether they're kind of in separate neighborhoods. I guess that's another indicator that might say whether you're going to respond to an immunotherapy. Rick, did you have any other questions?
When you said two drugs both targeting the same target, my question was, is that the same epitope?
Usually it's targeting a different spot of the receptor. So in the example of HER2, you can give Herceptin and pertuzumab for breast cancer. And most people thought that was a ridiculous clinical trial, but amazingly the patients did better. So that's kind of the model of that one that's FDA approved. But if you hit the same target, especially if that target is a strong driver for tumor growth, that hitting the same target in different locations on the target tends to lead to better responses.
Yeah, of course. You mentioned molecular profiling, and it's since I only imagine that your profiling involves genomics, not a gene expression or other aspects of.
So the ones we did at UC San Diego, so we took the tissue and then did the tissue testing and found it. We used to do them on Foundation Medicine because the deal with iPredict was it had to be Foundation Medicine tumor tissue testing to be on the study. So they had a 300 or more gene panel that they looked for different mutations that were characterized. We also did liquid biopsies for these patients. So the liquid biopsy, we used to go with Garden Foundation has one now, Tempest, all the major companies have one now. Dr. Kurzrock actually had some other things that she tested. There was a company called Omniseq. I think it's been bought out by LabCorp since then, but they looked at a whole profile of immune markers of response to other immunotherapies. So that one was actually better for matching to newer immunotherapies. The ones I was discussing, pembrolizumab, that's a PD-1 inhibitor. There's different immune checkpoints out there and many different drugs in clinical trials. So a lot of those drugs, you could get information from this Omniseq panel about immunotherapy matches. And then there was another one, which was a little not as well validated. There's a company called Paris, which had a protein profiling on markers of response to chemotherapy. So most of our patients didn't want to be on chemotherapy. They came to us for something new, but if we saw a match that looked good with chemotherapy, we wouldn't necessarily do it as the first line one, but we often do it on the second line one. Another thing logistically is that, so the liquid biopsy is just a blood draw. The other stuff is tissue testing. So you need to have enough tissue left from biopsies or surgeries. And we had to do it sequentially. So you send it to one company first and they do their work, then you send it to the next company and the next company. So usually these things come out over time, and you don't want to wait 4 months for all the stuff to come back before you start treating the patient. So usually we had the liquid biopsy, we came up with an initial recommendation, and then the foundation tissue testing came up with a second recommendation, and then the other stuff came in and we added to it over time. And sometimes you ran out of tissue after a while, or asked the patient, are you willing to do another biopsy? Some said yes, some said no.
Was the patient involved in how to allocate tissue for testing? Were they involved in that decision making?
Most of them just said we'll do whatever you want, and we felt that the foundation testing was the most valuable in coming up with match therapy, so we always did that one first. I don't think I ever actually asked the patient. The only tricky thing was sometimes insurance didn't pay. So with that discussion, they were actively involved. It's like with this one, you might have to pay a bill. Do you want to go forward with it? But we didn't actually ask them which one they wanted to do first, or if we just did order all of them.
So it sounds like you were looking at limited gene panels, whether it was through liquid biopsy or through tissue testing. Did you consider looking at whole exome or whole genome sequencing and how that might provide more insight for TMB that could be helpful in identifying treatments?
So the whole exome sequencing we often discussed when I was really actively with the precision medicine group. It was just coming out. It was newer technology, but generally, if we can't find any good matches for the patients, then we'll go to whole exome sequencing. I have one patient I'm actually presenting at tumor board today who he has one mutation. We targeted it, it's not working, and we need some more information. So him, we're actually sending for whole exome sequencing. The tricky thing with the whole exome is it tends to take a lot longer to get it back. It takes them a lot longer to do the testing. So anytime it takes it longer, that delays time to get a treatment to a patient. So we tried to go with the highest yield things first, and those were the limited panels, unfortunately, but it's always nice to have, Dr. Kurzrock always felt it's nice to have more information. I agree as well. So whole exome, I think we usually left that for if we needed more information, but it's important and it can give you additional information. But we just didn't do that many of them because we had a lot of good information from the test that we got.
Any other questions, Brian?
Yes. So I had a question about how long you monitored the cumulative effects of the drugs that you were administering. Sometimes you sort of get the shampoo effect, so toxicity can increase.
Mm-hmm.
And so I'm curious how you were monitoring that, for how long, and then also in terms of the durability of response, how long were you looking at the durability of response?
So for the toxicity, to start, we saw them once a week in clinic, and we got labs once a week. And when once we got to the level that we wanted for the drugs, where we thought we were at the sweet spot, where it wasn't too toxic, and we were getting some sort of response, at that point in time, we would space out the visits. We never let them go more than once a month without seeing us. And they had strict instructions to call us if there was anything that was off. But most of the patients we saw every other week.
And so you were monitoring them over like a year or just?
So most of the patients that came to us were coming from other doctors as a second opinion to do something new on the iPredict clinical trial. So we would monitor them from when we started therapy until they progressed and needed to go on something else. If we were the ones doing the something else, we'd continue monitoring them. But if they went back to their outside physician, then we gave it back to them. So it was from when they started till when they progressed and needed to go on something else. So if they were our patient, we keep seeing them regularly. If they were going back to their outside physician, we usually didn't see them back. And then you want to know about response.
Yeah.
So response got every oncologist has a different preference on how often they get scans. On non-clinical trials patients, I usually do the scans every 3 months. Dr. Kurzrock came from a heavy clinical trials background, so every 2 months we get a scan. There were some of the patients where we were doing separate studies about liquid biopsies. Can you see ctDNA rising over time? So some of the patients were monitored more closely. Some of them with biomarkers. We really like tumor markers. We tend to order all the tumor markers for everyone. Those were being done once a month. If we saw anything that concerned us with tumor markers rising or ctDNA rising, we did the scan a little earlier. If the patient had a new concern, symptom, we get the scans earlier. And then we did that until the patient progressed. And if they were our patient, we put them on something else and continue the monitoring. If they were out from an outside institution, they went back there, then they went and got their scans elsewhere.
Great. Just out of curiosity, was Dr. McKay part of this as a treating physician?
Dr. McKay was definitely on this as well.
Okay. All right. She's my primary medical oncologist as well as Rick's.
So she's a very, very, very talented and very, very smart lady.
Great. Thanks.
Eric, do you had a question?
Yeah, so I joined several minutes late, so maybe I missed this part, but I guess my questions are around access. What I hear you talking about is your experience of this through a clinical trial. And so I'm wondering, for me, like, is this type of route accessible by me now outside of a clinical trial? And the second part of that question is, as a first line treatment, I am still on my first line currently of just doing ADT hormones. I've not done, chemo or radiation and failed anything like that yet. And so sometimes I talk about these more advanced options and I find out, well, you can't get there until you failed something. So I guess I'm kind of wondering about access for someone like me right now.
Yeah, so the unique thing about iPredict, so iPredict was a UCSD-based trial, investigator-initiated. Most clinical trials, the company running the trial will pay for the drugs. iPredict was different. So all the iPredict patients had to go through insurance to get the drugs. Insurance denied it, we got it through patient assistance. So with regards to access, the access from iPredict would be the same as someone just coming in off of the clinical trial to get it. Currently at UCSD, my colleague, Dr. Shunei Kato, is still doing match therapy off of iPredict. He enrolls the patients on the PREDICT study and will do the match therapy there. So access is still available for match therapies for patients not on a clinical trial. And the drug acquisition, just because of that way that trial was put together, is the same for someone who's not on iPredict, who's just on the PREDICT protocol. With regards to your comment about first-line therapies, in my current clinic, I usually, if there's a good standard of care option out there, if you're doing well on ADTs, usually I recommend the patient stick with that. And if the first line fails, or if the next line doesn't look so good, or seems to have a lot of toxicity, then I would go looking for a clinical trial. The thing is, clinical trials, we got a lot of good data out of iPredict, and match therapy is looking really good, but it's still a study. It's not validated, it's not approved for use for everybody. So if your cancer has a good frontline treatment, I usually tell my patients to go to frontline treatment and then try the trial later. There are other cancers. There's one really bad one, adrenocortical carcinoma. Nothing works for that. So those patients, I will put them on a trial right off the bat. Just like, don't even bother with the frontline therapy. It doesn't work. I think it really depends on your cancer type of what's working. And then it's also the patient type as well. Some people want to be on something new from the get-go. And if they tell me that, then I'll refer them for clinical trials. If someone is willing to do the tried and true, first or second line, we'll do that, and then switch to clinical trials. I hope that answers your question.
Yeah, thank you.
Anything else, Eric?
Not right now.
Okay. Glenn Saban, you had a question in the chat there.
I was just curious, based on your experience and outside of the iPredict trial, those looking to access this type of management of their disease, there's most of the margin is still in actual drugs, delivery of drug, the actual drugs themselves, not necessarily the delivery of them. And I was wondering if those clinicians that were willing to provide the care that was recommended, were they within an academic setting or were they within a community oncology setting?
I think there was a mix. So a lot of I always joke because when I worked with Dr. Kurzrock, she would have me call the patient's primary doctor, even if the doctor hadn't referred them. And a lot of times I get yelled at for, why are you doing this? And even one of my own attendings from UCLA just ripped me up on the phone, like, what are you doing to this patient? She's responding, come on, leave it alone. But a lot of people from those academic medical centers have very strong egos and don't like being told what to do. So it was really a mix between academic medical centers, someone maybe a younger faculty who was a little more open to trying something new, and some of the community physicians, if they had nothing else to offer the patient, just wanted to help and were nice people and said, look, I'll do whatever you want to help out. So it was a big mix in who was willing to do it. And I never knew right off the bat who was going to be willing to help me or not. It was usually always a little nerve-wracking picking up the phone and calling them. And I just want to tell you what we recommended.
Yeah,
and even within Kaiser, sometimes they were very grateful. Sometimes they got really angry on the phone.
well, there is the economic issue that you have to be aware of, unfortunately. But I suspect that your list of collaborating oncologists has grown over time. Yes.
Yeah, so we had some groups that were willing to partner with us, but just based on where the patients came from, they were coming from all over the country and different parts of California. So it was rare that I got the same patient from the same doctor. Maybe Dr. Kurzrock would have a better idea, but I think the people doing precision medicine have grown, and she has a huge network of former trainees and colleagues throughout the country who are doing similar things with molecular tumor boards. So I think it's getting easier for people to find ways to access clinics that are willing to do precision medicine. It might not be exactly what we were doing at UCSD, but something similar. And as I mentioned, a lot of these patients would fly out to UCSD for a consult. A lot of things have changed with COVID, not necessarily in a bad way. So UCSD, before we couldn't do video visits, it wasn't allowed. Now we can do video visits. So if a patient's on a bunch of pills, I can order their labs at LabCorp request, which is nationally. I can see them on a video visit. I can talk to them, and we can kind of get everything together just remotely. And usually I would have them fly out to see me after scans because usually people want to see you in person after a scan, but some people, even that, they wanted to do remotely. So there's a lot more technology that's been opened up since COVID, just because we had to limit clinic visits for a while. And a lot of people living far away don't even have to come to San Diego too much, other than that first initial consult.
So
I
you're
just want to, okay, go ahead, Glenn.
I was going to say you're consistently you have the bandwidth to take on quite a bit more of patients seeking this type of guidance, this level of assessments and the delivery of the care.
So interestingly, based on the way I was rehired at UCSD, I can't actually do the precision medicine myself anymore. I have to refer them to Shimei Kato. but it's a logistical thing they're working out. But every patient we've referred to him, he's had no issue managing it. So, and I think they're hiring more people down there anyway. It's just that I was hired into a different group, so you have to be in the same group at UCSC to do certain things. But yeah, I think the bandwidth is still there. There's plenty of openings. So if there's more precision medicine clinics out there, and Dr. Kurzrock is seeing patients in Wisconsin now, so she has a clinic doing that as well.
Yeah.
So I think just the bandwidth nationally has grown with more and more people doing this precision medicine.
Yeah, in terms of your peers, across academia at NCI designated comprehensive cancer centers, are there others that are doing, what you're doing?
We always say at UCSG we're the best, but there are definitely other people doing similar things. It's not exactly the same, but there's other people doing things. And the track team for Target Cancer Foundation, they have the national tumor board where they provide the recommendations to the treating physicians. The patients can just enroll on that one. So any patient can be referred to the track team, and then we do our national molecular tumor board discussion and provide recommendations to the treating physician. So there's a lot more access for patients now than there was a couple of years ago for targeted therapy recommendations.
It's so great to hear. Thank you for the work that you're doing. It's really important.
You're welcome.
Yeah, I was just going to underline what Glenn was pushing on, which is we have found that the way around patients determining what their best treatment option is, maybe through a cure match recommendation of a drug combination, for example, and then the challenge of getting that access to that often means searching for a doctor who's willing to prescribe it. And so we're trying to almost build a group of doctors who are focused on that kind of personalized precision medicine treatment and willing to prescribe off-label as a kind of a workaround. And I think what Glenn was probing on, we often find that the dream team oncologists who are, the world recognized experts in those academic research center institutions are very conservative and really want to stay with the standard of care, really want to only do things that for which there's standard evidence. And so it's harder to find the doctors that are willing to do even what the patients are thinking is best for them.
The NCCN palliative path to hospice.
Yeah.
Just died. I'm going to see if I can get.
So I'm sorry, Rick, I've kept you on the background. What are you have several questions? Go ahead.
Hey, Mina. So I'm trying to, if you took on Eric, and I'll kind of read my question from 939. If you took on Eric or myself or any one of us, Mike, and you wanted to provide precision medicine guidance. So far, I've only heard genomic profiling. So, looking for basically driver mutations. Is there any, well, and, associated TMB, MSI. Is that enough to do? I mean, wouldn't you want to understand immune signaling in the tumor microenvironment, maybe to consider PD-L1 rather than just the, TMB or maybe IHC of TILs? Just curious, because all I've heard so far was genomics. And I think that
we were pretty heavily focused on the genomics for our precision medicine clinic, and I think in your question at RNA-seq and IHC. So most of the IHC, the PD-L1 is an IHC that's done. RNA-seq, the OmniSeq test was actually an RNA-seq test, and that one had all the markers of response to different immune checkpoint inhibitors. So a lot of these things were more experimental. So we get this data, but we would usually use as our first recommendation the things we had the strongest data for, and those are the genomic profiling. Some of these other things, the RNA-seq, we're definitely doing, but sometimes it was interesting. Sometimes we get a bunch of data back and results, and we didn't know what to do with it. So sometimes you have all this technology out there, you get a lot of information, and you have to figure out what do I do with it. And sometimes it also takes some time to figure that out. And I think with the OmniSeq, that was an RNA-seq for immune checkpoint inhibitors. That one we were getting more and more comfortable with, and as more clinical trials came out, we felt more comfortable. One example, the LAG3 inhibitors, that's another immune checkpoint outside of PD-1, PD-L1. Those have been recently approved for melanoma. So LAG3, that OmniSeq had the LAG3 on it, so we have a better idea of who will best respond to LAG3 treatments. So as technology improves, more clinical trials come out, we can use these technologies better. But there are a lot of other things beyond sequencing that we can do. We're just still trying to figure out what to do with this information.
Yeah, so maybe just related to that, Mina, so bioinformatics is just an evolving field. We talked a little bit about proteomics. We talked about RNA-seq. We've talked about spatial. I just recently tried to have an organoid built. I'm just curious, like how you're seeing, given Rozel's interest in deeper analytics, how testing is evolving, and who at UCSD is responsible for tissue strategy to enable the precision medicine that you're trying to do. Because from I think from my perspective, having limited gene panels really haven't been helpful for me to identify treatments. So you really have to expand the palette of tests. And so I was going after spatial recently and whole exome sequencing, and that just fell through, and that's a separate conversation. But who is responsible for evolving UCSD's tissue strategy to support this initiative?
You mean scientifically, or you mean who actually gets the tissue to the company?
Scientifically, or I would say scientifically, medically, who's responsible to say, hey, we could do better if we had for patient John Doe, if we had whole exome sequencing, if we had RNA-seq, if we had spatial proteomics, that's really how we're going to get a lot better with our treatment decisions. Who's responsible for that? Because what we see is a lot of amazing technology that's out there that can help us. The challenge is getting access to it. And there's a whole value chain or supply chain that's required to support the analytics and converting that analytics into testing strategies. And right now, I don't know who at UCSD is responsible for that. I just tried to play that role as a patient, and it was mind-bogglingly difficult.
Yeah, I think that there's no one person who's in charge of all of it. Everyone's doing little aspects. And there's a lot of segmentation. So with regard to the actually treating with the match therapy, Dr. Sumekata is doing a lot of that. A lot of the immunotherapy research, Dr. Sandeep Patel, he's the head of the clinical trials office. He's doing a lot of different things. He runs, him and Ezra Cohen are the ones that run they have a separate precision medicine tumor board based on immunotherapy clinical trials. They're called PIC. So they're the ones that recommended getting it's called Tempus XD, the whole exome sequencing for a patient that the limited panels weren't helpful for. But I don't know if there's one person that's kind of taking the charge of like, look, we got to go do this. I think you go to individual physicians, and I think Sume Kato, Dr. Sandeep Patel, those guys are really actively involved in precision medicine and immunotherapy research. So I think chances are you'll have some luck if you talk to one of them.
I guess the question is there a recognition that this is a need?
So I think there's definitely a need, but unfortunately in oncology, we kind of have to take small steps. We're still trying to establish the precision medicine and treating with matched therapies based on these limited panels. It's not even FDA approved to do what we did in iPredict yet. We're still trying to prove it. So we kind of got to prove that and get that approved and the standard of care, and then move on to these other tests. But you're absolutely correct. For for a number of patients, we do all these tests and we don't come up with anything useful for max therapy recommendations. And it's very frustrating to the patient and very frustrating to us as well that these limited panels don't work for everybody. And some of the limited panels, they expand over time. So the initial foundation panel was 200 things. Now it's over 300. There's an over 500 panel, but still a lot of times you don't find what you need. And even when they find something there that we think to drive to the tumor, sometimes we don't have a drug to target it. So it becomes challenging.
Yeah, the gap between like what's happening in biotech and what's actually delivered to the patient bedside seems to be getting wider. So, I was just at Illumina 2-3 weeks ago, and they're celebrating whole genome sequencing and what a huge triumph that is, and they're reducing the cost of it. Yet I tried everything to get access to whole genome sequencing, and I couldn't get it. So, and I had people within Illumina trying to help me to navigate that. And so,
I mean, it's something that I'm able to order, but I don't know if insurance. The other tricky thing is, will insurance pay? And if insurance doesn't pay, sometimes I feel bad because patients get stuck with a $5,000 to $10,000 bill. And some patients, that's not a big deal. Anything I can do for my health, others, that's most of their income. So
yeah. Well, I'll just log that as an opportunity with you, and Rick and I think both know Dr. Patel, so I can chat with him. Are you going to be at Ida Duchette's at her translational medicine conference symposium in April?
I don't think I'll be at that one.
Okay, all right. Okay, she's a UCSD clinical professor. Okay, that's fine. Thank you for your insights.
We're getting close to wrapping up the time. Rick, did you exhaust all your questions?
No, I had one other one that's kind of focused on average prostate cancer patient who we are deemed as typically having cold tumors. And so immunotherapy is a long shot because we don't have TILs in the tumor microenvironment. So when I was kind of hanging out with Ezra back in the human longevity days, oh, by the way, Chester, you look like Craig Venter, who, was the first to sequence the human genome, according to Craig, and, has that research institute on the corner of the campus. But anyway, Ezra was championing TLR 4 and 9 stimulation to try to heat up a cold tumor. Is there anything that you're aware of that has evolved along those lines, either with those agonists or other?
I think there are a lot of clinical trials that are ongoing with that in mind. How do we heat up a cold tumor? Some have suggested that some of these oncolytic viruses, put oncolytic virus in there that might rev up the immune system. There are a lot of people out there trying to answer that question. And when I was at Genentech for a year, the pharmaceutical company, there are a lot of people asking that question as well. How do we rev up these cold tumors so we can sell more of our immunotherapy? So there are clinical trials out there. It's still under active investigation. I really hope we can get something to make these immunotherapies a reality for the cold tumors.
Okay.
Yeah, any interaction? Ezra Cohen's our division chief, so he has many interactions with Ezra Cohen.
I didn't understand what you said. You had many interactions with Ezra?
Ezra Cohen's our UCSD division chief of hematology oncology. So any approval I need goes to he's my boss. So
Okay. All right. Yeah.
yes, we got to keep Ezra Cohen happy.
That's a good plan. He was doing a lot of vaccine work, or a bit of vaccine work. How's that going?
He has a lot of studies ongoing, lots of clinical trials. So with any clinical trials, some get some good data, some don't. I don't know the specifics of all of the studies, but he seems to be doing pretty well.
as a mention, and I'll stop talking, there seems to be a really good progress in bioinformatics community to interpret RNA-seq data, to deconvolve the estimates of different immune cells in the tumor microenvironment. I was really pretty impressed. There's additionally different signatures that can be derived from RNA-seq data. So I was really happily surprised, wow, just go on Bioconductor. And so I thought I'd pass that along as encouraging for that. When we use Tempus, I think it's a standard to profile whole genome or RNA-seq. They just don't share that data typically, which is, they keep it in there. So it might be helpful.
I think we can request the data from them. A lot of my colleagues like Tempus because if you ask for the extra data, they'll give it to you. But there's always a marker on something that's experimental, not for patient use. So you have to be careful with it.
And you can
Eric,
ascertain the same thing that OmniSeq does easily. But I applaud OmniSeq. I think they're great.
Eric, you had a question?
Yeah, so I had another question, kind of getting back to my access question again earlier. So I've had whole exome sequencing done on my biopsy slides. Came back with a couple mutations, and so I'm wondering if someone, like through what you're talking about, was matched to some kind of a medicine through that whole exome sequencing test. A lot of times, I guess I've found again that the medicines they match to are maybe approved for a patient who's castrate resistant, but I'm not in that case. And so I guess my question ultimately is around access again, is if I was went down this path and was matched with something, is there a way to get that kind of outside of standard of care because I'm not castrate resistant, right?
So generally, the recommendation when people come to see me for a consult for precision medicine, if they're on something that's working, I usually tell people to continue. If you're on something that's working, you don't stop it. And that was Dr. Kurzweil's philosophy as well. If you're on something that's working well for you, don't stop it, even if it's not the ideal therapy. And then we always get the matches together and have everything ready for if you were to progress. At that point in time, we would switch you to something else. So if you wanted to try the match therapy, it might be worth seeing someone in the precision medicine group to just discuss, the whole exome sequencing, what they'd recommend, and then have that in your back pocket for when you need it. But if you're on something that's working, I wouldn't switch it. That's the recommendation I tell all patients. Like, don't switch anything that you're on that's working.
That's fair, and I also think you get to another point is be working on your next step, right? So, all right, thank you.
I think it'll probably give you some reassurance to have your next step in your hands as well. That this is the next step.
Right.
Some reassurance, and to have everything lined up for when you need that next step. But hopefully, you wouldn't need it for a long time.
Great. Unless anyone else has any other questions, I think we're on time. It's top of the hour here, coincidentally or fortuitously. Anything else before we wrap?
Just have a very quick question. Do you have any kind of insight into the progression of the percentages of patients who can be helped by different modalities? So like the Foundation 200, I mean, not that many patients would benefit, but the 300, do you have any, is there any sort of curve on the percentages that are increasing as you go up this?
I don't have that data available to me off the top of my head, but we always feel that more information is better. So that the standard foundation 330/16 panel, we can usually find some sort of match for the patient. The 500 gene panel is actually for human sarcoma. So there tend to be some other alterations there that we are usually found there. We usually don't do the 500 gene panel unless we have a good reason to do it. But I don't have the exact data on how many more mutations we found on the earlier gene panels compared to the older ones. I'm sorry about that.
actionable mutations, of course.
Yeah,
And
there are a lot of unactionable mutations on those panels, but we often like to have the information.
And what about sort of the number of actionable mutations? I mean, if you do a whole exome sequence, which you could do a number of years ago and you can do now, the increase is got to be either because of the bioinformatics that you're able to understand more of the mutations as meaningful, or there are more drugs available for more targets. Is there any kind of insight as to the frequency with which targets are being drugged now so that more things are available?
I think in our precision medicine clinic, the vast majority of patients that came in, we could come up with a match therapy combo. So based on the liquid biopsy and the foundation medicine, once in a while, it was uncommon that someone came in and said, sorry, I have nothing to offer you. So there was almost always something we could do with that information. And usually they did not send us their information ahead of time for us to say yes or no. But once in a while, a patient will send us, like, is it worth me coming to see you with this information? And once in a while we say, hey, don't bother. We don't have anything targetable. You need more information. But the vast majority of patients that walk into the clinic, I had something I could offer them. But I think that's a large number.
And probably 5 or 10 years ago, that was not the case.
I don't know what was going on 5 or 10 years ago. Dr. Kurzrock would be better to answer that one.
Eric, did you have another question before?
Yeah,
we
my
wrap?
last question is purely logistical here. Mina, thank you for the call here today. It's really intriguing to me. I guess my question is, how would be the best way to connect with you if I wanted to talk more about like the match testing going forward?
Okay. Would seeing me in the clinic, or did you want to just an informal discussion?
I don't, probably informal discussion to start with.
I think Brad can give you my email address.
Okay, perfect. Thank you.
Did you get anything, did you get a cure match report, Eric?
I do have a cure match report,
I see.
and I have the whole exome sequencing through Caris Life Sciences, and I have something else from this group. What am I missing? I can't think all of a sudden.
Massive bio. I think
Yes,
you've got
there it
massive
is.
bio.
I had bio in my head.
Yeah. Quick question related to that. So, Mina, you have a group of, very engaged patients here. We're all, not all of us, but the patients here are advanced prostate cancer patients. We've seen a lot. 2 of us are being treated at UC San Diego. How can we help you and Ezra be more successful in what your objectives are?
I think you should probably ask Ezra what he needs for more research. I think they always say funds, but I think just being proactive, promoting things to other patients that are about match therapy and how important it is. And the more people are excited, the more we can get funds, we can get grants funded, and we can keep doing our research and keep helping out patients.
Okay. Great, thank you.